I think the short answer is there's many things that we can start to study now that were just not within reach even a few years ago. I think when we think back about the sort of how we're taught about genetics at school, we're taught about it from this sort of very Mendelian perspective, you know, a single variant giving rise to a change in a phenotype. But we already know that that's the exception, not the rule. Most phenotypes that we study, including most complex diseases, have a very complicated genetic architecture where huge numbers of variants, millions of variants, can contribute to changes in the phenotype. And this is something that we couldn't study historically. All we could ever do was go and look for the single variants having the largest possible effect. But now that we have these huge data sets, we can really start to study the sort of combinatorial effects of large numbers of variants and really start to study the sort of the true genetic architecture of disease and many other phenotypes. So I think it's really just changing our understanding of this mapping from genotype to phenotype at a very fundamental scale. I think another side of this is also rare disease is something we care a lot about. And the problem is that there are many rare diseases in the world, but each one individually is very rare. And this makes studying the genetics of these diseases incredibly challenging. But now that we have larger and larger cohorts, we can study the genetics of these very rare diseases. And this is incredibly impactful, not only for our understanding of basic biology, but also for how we care for patients and things like this. So in particular, if you have an ultra rare disease where a single patient is only known to have this disease, now you can do sort of matchmaking. You can find other hospitals with patients with similar genetic disorders, and they can kind of team up to try and find sort of strategies for managing the disease. And this is incredibly impactful on the life of the patients and their families.
Crucially, especially in our work, we're very interested in looking at variation across the tree of life and across diverse species. And well, when we're looking at sequences across diverse species, we're looking at the result of millions of years of evolutionary experiments. So we're just looking at what worked in a sense. And obviously, in a sense, we are going to be able to, if we're able to decode this information, to learn about the fundamental workings of fundamental biology. So in a sense, it's not surprising that we can learn from the variation across species to learn about human disease, because we're learning something about the fundamentals of biology. What really machine learning helps us is to bring new tools to precisely decode these vast amounts of data. What we're looking for are the patterns that have been preserved in sequence space, patterns that have been preserved and have been co-changing that are associated with functional constraints at the sequence level. And very often these patterns are really complex and the data is very highly dimensional. So machine learning models and in general, the new probabilistic modeling tools that we have access to just provide us with a new, just new set of tools to ask these questions. Yeah. Yeah, it's an interesting question, because I feel like when we started becoming interested in biology and we started being fascinated by biological systems, we initially naively thought that we would apply the tools that we were developing in physics to biological systems, but the reality was a little bit, it manifested in a bit of a different way. I think that what has been more impactful for us is the notion of model building. And looking at a system and thinking about how to express it in terms of a mathematical model to really critically assess what is the question I am trying to ask, what is the different ways of evaluating this question through a mathematical model. I think that that's really what's mostly carried over, and I feel like it has been very impactful for us from our physics training, and it's also something that we encourage now for our students and postdocs.
I mean, just to expand on that, as an undergrad in physics, you're always faced with the problem of, given some complicated situation, what are the assumptions that I can make that get to the essence of the problem, and what are the things I can kind of safely ignore? So there's these jokes about modeling a cow as a spherical ball in a vacuum, things like this. So oversimplifying the problem sometimes, these sort of lessons carry over to biology and to the sorts of work we do now beautifully, right? So we're asking questions about how do we extract signals of evolutionary constraint across the diversity sequences we see across life on Earth, for instance. And there's a very particular signal that we're looking for, but there's many other structures in the data that are obscuring that signal. So the question then is, how do we build a model that is sort of ideal for sort of throwing away or suppressing the bits we don't care about and really getting to the signal we want? And this sort of kind of almost art of model building is something that is a very, I think, physicist way of thinking about the problem, and I think something we have a nice training for. Yeah. Yeah, like Mafalda was saying before, when we look at sequences across life on Earth, it's sort of the result of the ultimate evolutionary experiment. And what we see when we look at patterns of sequence constraint on different evolutionary timescales teaches us different things about the underlying molecular biology of how a protein functions, or the role of that protein in a cell or in an organism. And so when we study the sequences found in a fruit fly, many things are staying the same and many things are changing. And this is sort of the perfect experiment in some ways, because we're saying that, okay, given that a fruit fly is not a human, which things stayed the same and which things didn't? And so, for instance, there's certain parts of a protein structure that have stayed the same across millions of years of evolution. And the same folds of the protein have been used and repurposed again and again and again for different proteins with different functions. But understanding those folds and what is required of the sequence for that fold to stay exactly correct for the protein to work is very important for our models. So seeing that, okay, the protein has the same three-dimensional shape in a very distant organism tells the model a lot about what's important for the functioning of the protein, and therefore what types of variation might disrupt the function of the protein and hence give rise to disease.
But in terms of critical thinking, I think that in this particular question, being able to tease apart what are the signals that are related to this question from other signals that might be important also that arise in this kind of data is very important. So it's always important when you build the models to really assess, am I asking the question that I'm really thinking that I'm asking, and am I evaluating this question to really capture the essence that I am trying to capture? And I think that the importance of critical thinking is particularly relevant and timely when we're talking about modeling with deep learning and AI tools in general. I feel like it's, in a sense, faster to do in silico experiments and almost easy to get some sort of answer. And it's very important to always ask yourself, is this exactly the kind of experiment that will give me the answer to the question I really want to ask? And that kind of sharpening your critical thinking tools is very, very important today, I feel.